So there's actually 3 papers, 2 from each. And then with those genes, I did similar to what they did in the article, I applied the criteria that they used for identifying which sites you might make a substitution at, or a stop codon insertion. And then I performed the same type of analysis that they performed, and generated a list for each protein of 5 locations where we might make a substitution. And then I've discussed with Kim too a little bit of the details on the different approach. We might use transcriptional control that I'll touch on briefly. And then Kim also has a few slides on, he's thought very deeply on mechanistically what we would do for the next steps of actually creating this plasmid and integrating it. So unless you have anything to say or any questions, I can go forward and kind of discuss a little bit more depth some of these genes that I identified, or these candidates, yeah, and how we might go about this.
Good.
Sure.
Sorry, Kim, can you mute yourself, because I can hear a lot of background talking? Okay, go ahead, Dylan.
So I just wanted to recap briefly. So from the Chang et al. 2024 paper, 2023, how they went about identifying essential genes and why it's important to find the essential genes. So of course, you identify the essential genes because if you insert the early stop codon that's recoded to have the unnatural amino acid inserted, in the absence of the unnatural amino acid, you don't get growth. And so the idea, of course, is that we would have this this controlled strain, or biocontrolled strain of a synthetic auxotroph, Cryptococcus, that would have the same property, or in the absence of O-methyltyrosine, you would see a loss of growth. So they were able to identify quite a few, just as a recap, quite a few essential genes, which you can see on the plot here from their, I think this is their supplementary figure 1, that they were able to get this approach to work with, and they used a few different criteria for identifying these genes. So they looked at, first off, they looked at the transcription level of each gene. And I think initially they started with low transcription genes that were essential. And that was kind of the criteria that they used. But of course, they're working with the benefit of working in SC, where a lot of genes are characterized. And so they're going off very deep literature in terms of what is essential and what is it. And there's no guarantee that what's essential in Saccharomyces is going to be essential in Cryptococcus, even though there are many genes that have the same name and similar functions. So I didn't look strictly at, say, CDC4 and well, they use CDC27 or CDC24, or any of the CDCs. In my case, I went back to the literature and I searched for genes that were reported as being essential. So if you knock them out, they can't be grown. And what I haven't yet done, though, is looked at the transcription level. I don't even know if that's available, but that's definitely a next step for me, is having identified these 6 genes. I'll try to find out whether there's any data on the transcription level and whether that will be a factor in terms of which genes we might select. So just still going through a recap here, what they did. And go further into how they selected sites within those proteins for substitution. They looked at the conservation, and they described it as the rationale for looking at conservation as sites where you can make tolerated substitution. The stop codon was the levels of conservation correlate with the essentiality of residues for protein activity. So the permissive residues might exhibit low conservation among protein homologs. And this are their conservation plots for their 2 main proteins, the CDC4 and the CDC27. And they mapped the conservation scores, just like in the figure there. And they observed this trend with a low conservation score towards the N terminus of the protein. And this isn't, I did a similar analysis with the 6 genes in Cryptococcus that I identified, and I didn't see exactly the same trend. But I just thought it was interesting to know that if that is something that we want to look for, maybe one or two of the genes that I identified will have this pattern. So are there any, that's kind of the end of the recap portion of that. Are there any questions? Their approach, what they did?
Mm-mm.
Okay. Um, so here's a summary again of the strategy that I applied once I had these target proteins from the literature. So of course, I obtained the sequence. I did a p-blast. I filtered using the criteria that they used. So that's throwing out everything greater than 90% sequence similarity, or between 70 and 90%. I removed redundant sequences using a tool called CD hit. So then I performed an alignment either two ways. One, there's this first tool that I used called Maft, and then the second way is kind of newer, which I like better because I can visualize it more easily. Was an R package called Prot Conserve R, though it's not. It seems more like it was somebody's project and isn't so published. So I might have to go back and look and try to validate this against the other approach and make sure that it's doing what I think it's doing. But
Dylan,
anyway, I was able. Yeah.
sorry, can I just ask, what are the 6 genes? Have you shown that already? Did
No,
I?
no, no. I'm about to go. I have so they are really quickly. They're HOM3, THR1, TRR1, RSA4, RPL39, and RPL22. Those
Okay,
are the six. Yeah.
the
So
6 that you
I'll
found.
just
Okay, go
yeah,
ahead.
yeah,
Yeah.
yeah. Um, so so I went through and I did the kind of analysis that they described, looking at each position, the conservation score. And just really briefly, I can't say that I know a ton about these genes at all, but here's the citations. And so I did the analysis looking at the conservation score, site-specific conservation score, um, having done the alignment, having filtered out, like I just talked about. And yeah, so I think if we were to choose, say, one of these genes, we have I have a pretty good sense of which positions we'd want to target first. Um, it's pretty self-explanatory. So if for example, for this pair here, these HOM3 and THR1 are part of the let's see, the homoserine and threonine biosynthetic gene. It's a
Mhm.
HOM3, and I think they're able to show that these are genes that are also important for virulence, but if they knock these genes out, the Cryptococcus is not even viable. So I think this would be a good candidate gene, one of these two, um, for first choice.
So
I don't know.
Dylan, you
Hmm.
have a substitution target,
Yeah.
and that one is by far the best because just recap that for me again.
Sure. So that's it has the lowest conservation score. So just what we're seeing in the plot here, if I can reorient you to, so we have these plots from their supplementary figures showing the conservation score per position. It's the same thing here, just plotted a little bit
I
differently,
see. Okay.
or different colors. Yeah. So the low conservation score just means that a substitution at that site is likely to be tolerated because among
Yeah.
homologous proteins, it seems like it differs so much that it
It
doesn't, that
varies. Yeah.
particular site, yeah, that residue doesn't matter to the function of the protein overall. So it can be substituted with the O-methyl tyrosine
Okay.
without, we can predict, yeah.
That makes sense. And in the THR1, I don't see, so it's more difficult to
Um,
find.
I'm sorry, so I just didn't highlight it,
Oh, oh, okay.
explicitly. So I have, well, you can't see my finger, but I have if you look under HOM3, I have the least conserved residues there.
Yes,
So that the red circle under HOM3, that's just pointing to that the
that right.
position 569. But I
Yes,
have the same ranking. So if you look, and you'll notice that these genes are different sizes too. So that might be a
yes.
factor, especially something to ask him about, especially if we want to construct these plasmids. There's some longer than others.
Mm-hmm. Mm-hmm.
Uh, but for the THR1, it also has a list. And if we look at in terms of, I mean, they're pretty similar in terms of what the minimum conservation score is. But
Mm-hmm.
like I was saying earlier, you'll notice that the low conservation proteins don't really all cluster towards the N terminus. I mean, I'll go back and
Yeah.
make sure that I'm doing this kind of analysis correctly, but it seems like whatever pattern they're seeing with the Saccharomyces essential genes we're not really seeing for these for whatever reason.
Okay.
But just something to note. And I
Okay.
think yeah, which I think I believe that they wanted to choose one near the N terminus because I guess the idea was if you get an early termination, you're less likely to produce a partially functional protein if it's
That
only,
makes sense. Yep.
yeah,
Yep.
yeah.
And so just one more thing. In the HOM3, it looks like you could have another site, right? If you kind of move across uh, toward the left, you might have a couple of options, right?
Yeah, absolutely. It looks like there are many
Yeah.
sites that have
Okay.
pretty low conservation. So we're not limited to this list of the five least conserved. I just I took the rank
Yeah.
or the
Yep.
positions. Yeah.
Understood. Okay.
Yeah.
Perfect.
And similar story with these two actually, and similar analysis here. I would think that um, maybe the TRR1. I guess it has it's a little bit longer, but it looks like it has some pretty low conservation score towards the beginning. So actually TRR1 might also be a good one to prioritize.
Hmm.
And here's the reference as well for for that. It's interesting. These uh, proteins are identified in course of looking for drug targets. They mentioned another one in this paper too, but I couldn't find any enough homologs to do the kind of analysis here for it. It's something to note. And then finally, these are, I guess, of the last two of the six that have definitely in the literature been uh, explicitly called essential. Is the RPL39 and RPL22. And I guess the RPL39 is fairly short, so maybe it'd be easy to work with. But same story again. I was able to do the kind of analysis that they described in the paper to identify potential positions for substitutions. One
Nice.
thing that I haven't yet looked at though is I might want to rank these by just how many homologous sequences I could find. I haven't done that yet, and that might bolster the confidence of it. So just looking right now at the RPL39, the fact that so many of them are on the same level kind of to go back and check, but it might not have had too many similar homologous sequences, and that's why you're seeing so many
Mhm.
at the same level of of conservation. So I might want to go back and factor that in as well. Um, and then I looked at this for a bunch of other proteins that aren't explicitly called essential, but I think some of these are familiar, like the capsule protein RAS, CDC4 and CDC24, like we were talking about last time. I did the same analysis in all these as well. Uh, yeah. And
Okay.
so I guess this brings up another question: is do we need genes that are definitely essential, or can we pick ones that are just known to have a strong role in virulence? I guess that's something to think about. Well,
Well, I would think you want it gone, don't you, Jamal? You don't want it lingering around, right?
Yeah,
Maybe,
we wanted that. So I think, yeah, certainly. I'm not even sure you know, I'm not even sure we actually want to target virulence because that might play a part in its ability to get into the brain. But certainly viability, if this is
viability,
something
yeah.
that kills the bug, then yeah.
yeah. So I think hopefully all the six I mean that was um, my sense as well. I think that the six preceding this slide here should all work for that if
Mhm.
one completely uh, non-viability after the O-methyltyrosine or the inducers we'll talk about in a second are gone. Um,
So Dylan, one more thing.
mhm.
So if you now try to find the optimal one, as you were saying, you would want the mutation or the target gene, sorry, nucleotide or amino acid to be close to the N terminus, right? So that kind of that removes some of these, right? So if you're focusing now what's closest to the N terminus, and then maybe what is a gene that is shorter versus something that's very long. It might be easier to work if we're cloning it and all of that. So maybe those are two other factors that could help you narrow down which ones you can concentrate on, I think.
Yes, that's true. Though I did notice that some of the shorter ones, it was maybe this is not a general, but it seemed like it was harder to find homologous sequences for some of the shorter ones.
Mm,
So I'll try to
okay.
balance though. Try to
Yeah.
find the shorter one that have low conservation towards the N terminus. Yeah,
Yes.
definitely
Okay.
prioritize.
Hmm. Right. Yeah. I mean, I wonder whether, Kim, you raise a good point. I know doing it in crypto can be tough, but when would it be possible, I guess, as you're manipulating it, we could express it in Saccharomyces, and then when it's ready to go, then we would take it out and put it in crypto at that point. You know what I mean? Be would that help? Yeah, they are. They are. Yeah, right, right. Yeah. But there might be enough homology that they do, and if you could work in the background of Saccharomyces, that would make things a lot
Mm.
easier for sure. Yeah.
The plan is to screen in Saccharomyces first, essentially.
Yeah, in a way, that would be the idea, right?
Yeah.
I think that that could just simplify the whole process if we could get that to work. And we've got all the plasmids for Saccharomyces. We've got high expressing ones, low expressing ones. So, and it really is easy to manipulate. Yes, that's right. That's yes, exactly. Yeah. Okay, good. So far, so good, Dylan. This looks really cool.
And then Kim and I were discussing a little bit, and I think Kim is especially keen on this. The second approach mentioned in the article, sort of the backup, where they used a promoter on a separate orthogonal essential gene, um, and use that as a second layer of control. So we're thinking a little bit about this. I think there are some the separate hurdles, but definitely something to look into. I identified. Well, I mean, one of the hurdles is obviously in the host we don't have as controlled an environment in terms of what inducers might be around. So we can't use some of the inducible promoters that might be convenient in fermentation. Um, but I think that there's, for example, this one article that I found is 2024, fairly recent, again in Saccharomyces, but using, I think they identified 5 different promoters that they were able to use some synthetic promoters, in fact, that they're able to engineer into Saccharomyces, and one of them was triggered by doxycycline. So I imagine that maybe it'd be possible to load doxycycline into PLGA and use that as an inducible promoter. If we're able to get this to work in Cryptococcus, it could be the main or at least a backup strategy using the transcriptional control. I guess one note of caution is I believe that in this paper here, I think they said that they were able to achieve a 60x differential in expression with with some of their best promoters. I don't know if that's good enough
Mm.
for what we might want to see, but I think certainly as at least a second line, it might be something to look into, or at least something to mention in any proposal as an alternative approach. I think. Um, so I believe that yeah. Next, I have a few slides that Kim just sent me. I don't know if Kim wants to. I'll just switch it when you tell me to. I'll turn off my mic really quick.
Mhm, mhm, mhm. Mhm. Previous slide, the one before, yeah, there you go. Yeah,
Yeah.
it makes sense. Mhm.
Yeah, there's escape
Escape.
control. Yeah. Okay, thanks. Let's see if I can go back. I just wanted to mention too, I believe some of these more recent articles, this one, the 2020, um, I think, Kim, maybe we should look at this one together because they go into some strategies for how we might, this because they had to do some of this engineering in Cryptococcus for this. So it might be a good resource. I just
Yeah,
wanted to
yeah.
mention that. Yeah. Um, and then that's about it. So I've kind of this mostly blank slide here because obviously I want your input too and your feedback on what we've presented so far um, on the next step. So of course, I'll continue to talk to Kim about the finer details of how we might do the and Andrew too, obviously, but whether we want to start ordering things or writing a proposal or something of that nature.
Well, what do you think, Jamal?
All of the above. Um, in terms of the right strategy, this is you guys' area of expertise. And so we will heavily rely on you to sort of advise us or, um, as you just have on, which is the best strategy. I I am, I hear Kim out on the galactose strategy, but
Yeah.
I still think that the a natural amino acid is something that would be really novel and really cool
Hmm.
in physiology. I don't think I've come across anything like that in physiology itself. And I think the innovation there, along with innovation of putting particles on a fungi for drug delivery, I think is gonna kind of blow their minds. But 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 again, yeah, I rely on you guys to sort of best engineer that vector into cryptococcus for lethality. So
Well, Kim has he has dealt with pretty sophisticated and complicated cloning in the past, and he's gotten it to work. So I have a lot of confidence in what Kim can clone, and I think with Dylan, I think the two of them can really make this work. I think it's really exciting, Kim, and I do think that there is going to be, hopefully, with the papers that you showed, Dylan, some of the work that they did with crypto with some of these genes might be helpful to get some of that information out of their methodology to see if that maybe facilitates some of what we can do. And as I mentioned, if we can work a little bit in the background on Saccharomyces, that might also make things a little bit easier. And the other option too, Jamal, something we've been doing a lot is instead of just cloning things ourselves, we're sending it out to companies to kind of do it for us because they can do it quickly and it's gotten pretty cheap. So
mhm.
Kim, if at any stage you find that that can facilitate the process, we can maybe also do that. So I think that approach is worth pursuing. And then, whatever you need to make it easier or to facilitate it, just do what you need to do, I think. What do you guys think?
Um,
What do you mean both approaches? The galactose approach? Well, Well, but as Jamal mentioned, I mean, part of the attraction with a natural amino acid is that it is pretty innovative. I mean, galactose inducible is pretty old. I
Yeah.
mean,
um,
it's I don't know.
so
Yeah. So
So the other thing that we want to consider, Kim, is that if we were to do both, it means we have to put both a natural amino acid and galactose into particles and put these particles in the surface of the cryptococcus. And our main goal here is still to deliver drugs via these particles, not to sort of kill the bug. So what I'm saying is that we have limited sort of capacity for
for
both.
which you can load up. Yeah, that makes
now
sense.
one other thing that I thought we might want to emphasize too in the strategy is it is taking a pathogen and uh, introducing a biocontrol strategy for the pathogen. So I wonder um, if part of the appeal for working on in introducing the synthetic amino acid is that it has ramifications for safety as well. If you want to work on a strain that, like they mentioned in the original article, I mean, their purpose was they were trying to prevent it to get into the environment for industrial espionage. I think they mentioned over and over. But I think obviously it's interesting as well for just safety for working with the pathogen too. So I think that's why I would be again with galactose. I mean, maybe it can come across galactose in the environment as well, but it maybe a different inducible promoter, or something like doxycycline or something. But yeah, that's
Yeah, I
something
would
else that
agree.
I was
I would
thinking.
agree with that completely, though. I mean, galactose is produced uh, based on sort of metabolic stresses within the body. So
Um, but anyway, I'm excited to work on this, and I don't know what the first thing that we might want to if we want to start doing what work, what exactly that would be. But I'm very excited to
so
actually
Angie,
get my
this all depends on how honestly, it depends on how an aggressive of an aggressive timeline that you're open to.
Um,
Uh,
well, you're looking to submit, I mean, you're looking to submit a grant, right,
mhm,
Jamal?
mhm. Yeah.
And
So I'm hoping to submit in June, which would be basically a month from now.
a month
I
from
don't
now.
know if you guys can get any preliminary dates in a month. Might be a little bit of a challenge. But that being said, I do have continuous submission, so it can actually go in a little bit later than a month from now.
Yeah. Um,
Yeah, yeah, definitely.
I mean, yeah, because it would speak to
Yeah,
feasibility, right? So even if the
definitely.
whole thing is not completed, but we're showing that what we're doing is working, I mean, I think,
So
and it's
the goal,
preliminary.
sorry,
That
sorry to cut you off,
No,
Angie.
go ahead, go ahead.
I was just saying that the goal of the grant, Kim, would be to put the full thing together, right? But if we
Yeah,
have feasibility data showing that, hey, this concept works,
yeah,
right? Um, then we're golden.
yeah,
PM, like feasibility data can also be in just the Saccharomyces,
yeah.
right?
That's
We've
true.
identified these this different homologs that target in these different genes, and we put them in Saccharomyces, and you know we put in the plasmid, and without the unnatural amino acid, methylated amino acid, these things die. That's
Yeah, exactly, exactly. And all the in silico analysis, Dylan, that you've done, finding the six genes, the region that would be of interest, all of that would be part of the
preliminary data right there.
preliminary data. So that's all great. Yeah, for sure. Yeah. Do you think, does Addgene have that, Kim, or do we? Okay, okay. And maybe, Dylan, the other thing that you can do is maybe just kind of start writing it up a little bit. Would that help, Jamal, if Dylan just sort of starts slowly writing down, what the procedure would be?
Yeah,
Uh,
so how I'm envisioning this is in R1 with three different names. We're hoping to target pancreatic cancer, and so I'm trying to work with another assistant professor who's also a pancreatic um, cancer surgeon here at UF.
mhm,
Uh, we're hoping to get some preliminary data just using gemcitabine loaded nanoparticles
mhm,
for what would be aim 3.
yeah,
In one, we're loaded. We have a lot of data. Clinton's done a lot of work showing accumulation of the bugs in the pancreas via
Mhm.
luminescence
Mhm.
in vivo imaging. And then we've also done a lot of work, looking at, seeing we can put these things on the surface, load them with drugs, the extent to which we can load those drugs. And then we've also done release kinetics of dyes. All that to say, we have for, how I envisioned it, aim one would be sort of engineering the fungal nanoparticle hybrid carriers, and that would be sort of our responsibility. Aim two, and we have a lot of preliminary data for that. Aim two would be essentially engineering the fungi to be safe, right, via sort of genetic manipulation. Um, and yeah, like Kanji said, all of the in silico work they just did, Dylan, that is preliminary data right there. But if we could actually get some additional preliminary data showing this is effective in blunting the viability of the bug, then yeah, we have the concept, and we should be people should be excited about two novel concepts, and then applied to pancreatic cancer, something really difficult to cure or to treat. Yeah, this could be a really good grant.
That sounds awesome. Yeah, I can get of course I'll send you all the data and what I have for each of these genes as well. And hopefully if that helps put it together into a figure for the grant, then we can get that to you. Yeah.
Sounds good. Mhm.
Yeah, so I think I still have Box. Clinton might not have Box, so that's the only problem.
Ah,
We use Dropbox here at UF. But I don't know. I can maybe act as the go-between. I don't have my UC Davis email anymore.
oh, but I think we can invite you, right? We can send out an
You might be
invitation,
able to. Yeah, yeah, yeah,
and
yeah.
then Clinton and
Yes,
Jamal can both
I
access
believe so.
the
Yeah,
box.
yeah,
Yeah, yeah, yeah. That shouldn't be a problem.
yeah. So if we set up a folder, then I could probably put together maybe a framework for what I envisioned for this grant,
Okay.
and then we can just start filling different pieces.
Yeah, that sounds good.
And I'll keep the tentative deadline of June 5th, but if you guys can't make that, we can push it back to July 5th or even as late as August 5th.
Okay. Sounds good.
All right, I'll set that up with the RAs here, the CGs here, and Angie. I think they'll probably be emailing you shortly as well.
Okay, that sounds good. All right. Anything else? Anything else you want to say, Dylan?
No,
No?
Clinton, you want to share some of your work just so that did you share it last year, the last time we met? I can't remember.
Not really. I didn't know how freely we were sharing any information.
We're putting together a grant, so we can share.
Okay,
And
cool. Sure. Uh, okay, I sent a request to
yes.
share.
we got like 16 minutes, so I might as well use the time.
Yeah.
Okay.
Dylan, I think you have to make Clinton host.
Okay.
So if you make him co-host or host, then Clinton can share his screen.
Okay. Do you have that permission?
Yep.
Perfect.
And Dylan, you mind sharing that this PowerPoint with us as well?
Yeah, definitely. I'll upload that as soon as
Into the
I
folder. Yeah.
Just to confirm, you guys can see my screen?
Yes, beautifully.
Okay, perfect. All right, so yeah, all of this work is hopefully going toward developing this novel drug delivery platform that could be leveraged for uh, a number of different indications, one of which Dr. Lewis mentioned with pancreatic cancer, depending on the route in which we deliver. But at least our initial idea was to get these to the brain and further improve drug delivery across the blood-brain barrier. So I will skip a bit of the motivation that I'm pretty sure we're all aware that lots of people are afflicted with central nervous system conditions, and they vary in terms of need for effective therapeutics. And the biggest challenge with treating these different diseases is the fact that the blood-brain barrier exists and prevents about 98% of drugs from actually crossing. We do know that it's not impossible to cross. There are a number of different strategies to do this that a lot of molecules or even pathogens have been able to execute. And we know C. neoformans is sort of professional at navigating the central nervous system in that it can execute 3 distinct mechanisms. So based on the fact that it can execute 3 distinct mechanisms, we're primarily interested in this Trojan horse delivery mechanism that has been observed with C. neoformans. And in particular, leveraging vomocytosis, which is this non-lytic escape mechanism that the pathogen executes to liberate itself. So yeah, the big question that we're trying to answer is whether or not we can leverage vomocytosis and the CN to improve therapeutic delivery to the brain.
Yeah, and again, do you have this distribution data in here, Clinton, or?
Yes, I do. Yeah. So the big idea here is that we could overcome the challenges associated with standard and effective drug delivery and that they're unable to cross the blood-brain barrier by simply loading drug-loaded nanoparticles onto the surface of C. neoformans and leverage its natural neurotropism by way of one of the 3 different mechanisms, but primarily through this immune cell hitchhiking mechanism. Okay, I don't think Zoom likes PowerPoint right now. Okay. So yeah, what I've been working on for the past 2.5 years or so is this attachment process. So I looked at using PLGA, polylactic co-glycolic acid, super common biomaterial that is clinically used in a lot of different products, in particular biodegradable sutures. And in terms of drug delivery, it's very advantageous because it's very tunable. You can engineer it to perform in any sort of capacity by simply adjusting the ratio of the 2 different monomers or adjusting the molecular weight, and even introducing some other moieties that would make it more sensitive to drug release. And so with this approach, we tried simply sticking the nanoparticles on by adsorption, so using intermolecular forces. And we also looked at establishing a covalent bond between the nanoparticle and the CN surface to yield these particle backpacking fungal carriers. So to make the particles, we have this classic process where it's just a simple oil and water emulsion to create these nanoparticles. They're quite small in size. Oh God. Okay, here we go. And they're quite monodispersed according to the polydispersity index. And in relation to the core diameter of the cryptococcal cell, we have several orders of magnitude difference. So theoretically, these particles should be able to fit onto the cell surface. But things get really tricky when we look at the surface charge of these components. So we know that CN tends to be negatively charged, and our ordinary PLGA nanoparticles are also quite negatively charged. So through basic physics principles, we know that things that would have like charges are likely to repel each other. So I decided we could introduce this method of coating the surface with a cationic polymer or material to basically leverage some electrostatic interactions between the nanoparticle and the cell. And so I did some literature search, and basically polyethylenamine was the lead candidate for coating the cell surface, particularly with yeast. This was done with SC previously, and then I eventually implemented this with C. neoformans. Okay, so I had two different approaches. I think I'm just going to focus on this first one for now, which is this layer by layer approach, where you basically take the negatively charged cell surface, coat it with polyethylenamine, which is the cationic polymer, and then you do a different mixture with these PEI coated cells with the negatively charged nanoparticles and hopefully yield these particle backpacking fungal carriers. So it's a pretty facile process. God. Okay. So I literally just take a
I think it's
I
good
take
to
a colony. Yeah, I take a colony, grow it overnight. I literally just mix the cell solution with a solution of PEI, wash away the excess, and then do a second layer assembly with the nanoparticles. All right. Seems like if I use the arrows, that's better. So yeah, before I attach the nanoparticles, this is what the pellet tends to look like. It's this off-white yellowish color. And then after I execute that that process, I do see the faint nanoparticle color, this somewhat turquoise or teal color after I've attached the nanoparticles. And using flow cytometry, I was able to confirm the successful attachment of these nanoparticles. So if we just have the PLG nanoparticles and unmodified CN, we get very minimal attachment. But when we coat the cells with that cationic polymer, nearly every cell has at least one nanoparticle attached.
And these are unloaded nanoparticles at this point. They're just green on their physical property.
Yeah, so we conjugated a dye to the PLGA, so right now it's technically unloaded.
Okay.
But when I first started doing this, I was loading a molecule, a dye into the nanoparticles, and we saw basically the same thing. But for, I guess, better imaging, it was easier for us to just use the conjugated PLGA. So yeah, using this approach, I was able to somewhat estimate the number of nanoparticles we can get on the CN surface. So if I attempt a 200 to 1 nanoparticle to cell ratio, I get about 45 nanoparticles per cell. I have no better frame of reference for this since it's so novel. So the efficiency is about 22%. We don't know if that's effective or not. But it is what we have thus far with this approach,
We've also
at least.
increased that.
Yeah, I've increased it by doing a completely different assembly. But yeah, for the sake of time, I started to look at how stable these particles are on the surface. So I basically incubated them with serum proteins. And so the idea is that if the particles are unstably attached, the proteins could easily knock them off, and I should be able to visualize that through fluorescence measurements. And I was able to confirm that when we attach these nanoparticles and the PEI, they remain viable compared to our wild type CN. Pre and post serum incubation, they remain quite stable in terms of the number of nanoparticles that or in terms of the number of cells that have nanoparticles attached. And then when we look more closely at the medium fluorescence intensity, we don't see any drastic drop-offs in terms of fluorescence either. So the nanoparticles tend to stay bound to the surface after the serum incubation. So that was promising for us. And that's with adsorption and with conjugation as well. But most critically, we wanted to demonstrate that when we attach these particles, we don't see any drastic changes in its ability to undergo vomocytosis. So we compared two different ratios, 50 nanoparticles to 1 cell and 200 nanoparticles to 1 cell to our unmodified or wild type CN. And we don't see any statistically significant differences, but we do see this trend, at least through two biological replicates, that there's this upward trend that once you attach the nanoparticles, it seems to become more hypervomocytic, so to speak. And then the next step for us was to look at where the CN accumulate using different delivery routes, and we wanted to maximize delivery to the brain. And so we tried intranasal, which is I guess the more natural mechanism by which CN infects the host. We tried intraperitoneal, IV, sub-Q, and intramuscular injections, and we investigated each of these, its accumulation in each of these organs. And so, yeah, we used IVUS and bioluminescence to execute this. And pretty interestingly, we were able to determine the delivery route that maximizes accumulation in the brain. It turns out that intravenous delivery works best for that. But we did get some other exciting data that could serve as implications for other things we can target. So intraperitoneal delivery seems to result in higher accumulation within the pancreas. And then with intramuscular delivery, we see this accumulation within the neighboring or draining lymph nodes, which was exciting for us to see. And pretty unsurprisingly, with intranasal in healthy mice, majority of it remains in the lungs over the course of 3 days.
And is that the window, 3 days, that you think we'd want to be looking over in terms of if we want this strain to last?
Yeah, essentially.
Yeah.
I think we want to, have it do its thing, accumulate in whichever respective organ, and then
Yeah, so that is the bulk of the data that I have, but I have been able to improve, or no, actually, I forgot that I included this. So we took it a step further and looked at if we attach these nanoparticles, how well do they distribute to the brain? And so using this layer by layer approach, in addition to click chemistry, just to really ensure that the particles are stably attached, we conjugated them and injected them IV to some healthy mice, and we excised these tissues and used bioluminescent imaging. And using flow cytometry, I did a bit of quality assurance just to demonstrate that the FDCs or that the cells did have nanoparticles attached. And yeah, through the bioluminescence data, we were still able to see that the cells are capable of infiltrating the brain, which
this was just intravenous, right?
yeah, this was intravenous delivery. So the one that we identified as maximal for brain delivery.
Mm-hmm. Very nice.
Yep. And this is just compiled data from the 3 replicates that we have thus far. But yeah, pretty excitingly, we're able to show that we can get the cells into the brain. We're still working out some ways of showing a co-localization of the nanoparticles and the cells, just to really hone in on the fact that we need the fungus to facilitate nanoparticle delivery.
Mm-hmm.
But thus far, we're excited by what we're seeing.
And Clinton, the strain of crypto that you're using, is it just like an H99, just a typical lab strain?
So it is an H99. Well, it depends on which experiment. So for the animal studies, it is the H99 serotype, but it has been engineered to express luciferase.
Oh, right. Okay. Yeah.
But for the other like ex vivo or in vitro studies, I've been using just the standard H99 strain.
Okay. Well, the data is very promising. Yeah. Sorry, go ahead, Kim.
So yeah, I have done that. I don't have the data in this slide deck, but one of my undergrads over the last couple of weeks has demonstrated that we can attach the nanoparticles to the acapsular strain because well, for one, we know it can get to the brain and it ends up dying off. Um, and we also know that acapsular cells are also negatively charged. So we figured that that would be another way to demonstrate that we can attach these nanoparticles. Yeah. Is that STUD? Okay.
No,
No, so we
great
inject
idea.
it on its own. Yeah, so as you mentioned, we're trying to figure that out, but I think the evidence that's out right now would suggest that the Trojan horse mechanism seems to be the most prominent, just based on the different controls that people have tried in their studies. Knocking out monocytes seems to very significantly decrease the amount of CN that infiltrates the brain.
Yeah, I mean, certainly we know that transcellular contributes, that's for sure. Whether it's more Trojan horse or not, I think that's debatable. I don't think that's been clearly worked out.
Yeah, I to be quite honest, I
Yeah, we don't care how it gets there, as
Yeah,
long as it gets
exactly,
there.
exactly, right? I mean, and I guess you haven't looked to see, how well the mice tolerate this, or is it immunogenic in any way? Do you track anything like that yet, or you're just not there yet?
We're not there yet. That's something that we have thought about.
So
Um,
anecdotally, I mean, you can talk about what happens, I guess, and you guys have
yeah,
probably seen this as well, right, Angie?
Yes.
Yeah.
yeah, we've noticed that at least from a survival standpoint, the mice who were injected with these fungal drug carriers tend to survive slightly longer,
Mhm.
um, maybe within like a day or two of each other. But uh, yeah, it also depends on the dose that we
Right.
deliver with these strains.
Yeah, that makes sense. Yeah. Okay. Nice work, Clinton.
Ah, thanks.
Yeah, very good. All right.
Great.
I need to move on. I've got another meeting.
Yeah.
So, yeah, I'm going to take off if you guys are going to continue talking. That's all good. I guess we'll touch base again in a few weeks, maybe see how things are going.
Yeah, I think we can uh does 2 Mondays from now work for you guys, or do you need some more time? Maybe 3 Mondays from now?
That brings
Mondays,
it.
I think that should work for
Yeah.
me.
Two or three.
Let me make that
First
Three.
Right. 3 Mondays from now. I'll send a hold in your calendar, and yeah,
It
3 Mondays
was,
from
but
now,
yes.
we have our normal meeting time. But anyways, yeah, it's fine. 3 Mondays from now,
Okay,
we'll be in
that's
touch. And
perfect.
Angie, I think my CG should probably be getting in touch with you
All
shortly.
right. That sounds great.
All
Thank
right.
you so much.
See you. Thanks,
Right.
Dylan.
Bye.
Thanks.
Thank you. Bye.